We're very excited to have Professor Sang Zhong from UCSD. And Professor Zhong is a professor of bioengineering at UCSD. And previously, he was at the University of Illinois at Urbana-Champaign for about 7 years. And he received his degrees from Peking University and Harvard. And after a short Stanford visiting scholar position, he started his faculty positions. And currently, he leads the team for systems biology at the UCSD. And he received many awards. I'm going to name a few. One of them is NIH Director's Pioneer Award, NIH Catalyst Award in Diabetes, and NIH Director's New Innovative Award, NSF Career Award, and Alfred Sloan Fellowship. And his 8 previous trainings actually now contributing to tenure track positions. And that's actually very impressive. And without actual further ado, I'd like to turn to Dr. Zhong for this exciting seminar.
Thank you very much, Ahmed, for the very nice introduction. I'm glad to have this opportunity to share with you some of our interests on studying the spatial distribution of RNA. And I wish to speak today to share what does RNA do on chromatin and on cell surface. So the long-term goal of my lab is to understand the functions of different parts of the human genome. While we now have good information about 10% of the human genomic sequences functions, including their roles as coding or non-coding genes, their roles as regulatory sequences, as promoters and enhancers, there remain to have a large fraction of the human genomic sequence that do not have its all functions annotated. So how we go about to study the functions of the vast majority of the remaining genomic sequences of the human genome. So one striking observation that struck me was that more than 60% of the human non-coding genome is transcribed into RNA. So we thought that perhaps by studying and identifying the functions of these transcripts, we could start to have a view of the functions of the underlying genomic sequence. And we hope to identify the functions by studying the localization and interactions of this RNA. So I hope to share 2 research directions from our lab. So one is on chromatin-associated RNA. These RNA attach to different parts of the genome, and I wish to discuss whether they serve as a type of epigenetic regulatory material. The other type of RNA is stably attached to the cell surface, and I hope to use the second half of this seminar to discuss whether they represent a type of cell surface molecule and perhaps be responsible for signaling. So for the first type, chromatin-associated RNA, so these RNAs are transcribed from one gene, and they sometimes travel and attach to another gene on the same chromosome, and sometimes they somehow travel and attach to the genomic sequence of another gene on a different chromosome and mediate transcription regulation. So a few of the already discovered pathways for this RNA-mediated transcription regulation, including, for example, the RNA help to stabilize transcription factor chromatin interactions. The RNA contribute to modify the histone decorations near the target gene, or perhaps the RNA help to stabilize certain spatial arrangements of the underlying genomic sequences. So our main questions when we started to investigate this type of RNAs are as follows. The first is, what are the chromatin-associated RNAs? Previously, we've seen a few examples from literature, but in general, what genes or intergenic sequences give rise to these CA RNAs? The second question is, what do they say RNA bind in the genome? Where are the genomic binding sites or interaction sites of each chromatin-associated RNA? And the third question is, do any of the binding correlate with transcription levels of the target genes? So to study, to ask these questions from an unbiased manner, several postdocs, including Dr. Wei-Xing Wu and Trinh Ren, have continuously developed and updated technology, which we call MARGI, Mapping RNA Genome Interactions Technology. The more recent version is called in situ MARGI, iMARGI, that is meant to systematically discover in an all versus all manner all the nuclear transcriptome versus genome interactions in one experiment. So the idea of this technique is that suppose on our genome, on different parts of our genome, there are different RNA molecules attached to different parts, and we wish to stabilize those interactions by cross-linking the cells. We fragment the genome and sometimes also the RNA, so that in some of the fragments, we would have a piece of DNA and a piece of RNA that is stabilized together. Then we wish to convert this DNA and its proximal RNA into a unique piece of DNA that can be sequenced by DNA sequencer. The way that Wei-Xing and Trinh did this was to introduce a linker that is ligated to one end of the RNA, and through another round of proximal ligation, if there is DNA in the spatial neighborhood of this RNA, then this linker will be ligated to the proximal DNA fragment as well, thus forming an RNA linker DNA chimeric sequence. Then in a few subsequent steps, this chimeric sequence is converted into double-stranded DNA suitable for typical next-generation sequencing. With paired-end sequencing, we can actually know which end corresponds to the original RNA and which end corresponds to the original DNA, and thus use a read pair to infer a particular interaction between one RNA and its target genomic sequence. So below here is an example of inferred interaction between MALAT1 RNA, whose gene is on chromosome 11, to this target genomic region near this KTN1 gene on chromosome 14. So here, every row is a read pair where the RNA end is mapped and shown in red block, and the DNA end is also mapped, shown in a blue block. In this case, we see that all these read pairs were mapped to the RNA of the MALAT1, and they are associated with the genomic neighborhood of this KTN1 gene. And these read pairs are used collectively to infer interchromosomal interaction between the MALAT1 RNA to the genomic sequence around this target gene. So this type of data in a genome-wide is typically now summarized into RNA versus DNA contact matrix. In this matrix here, what's shown on the y-axis is the entire chromosome 1, starting from its start position to its end position, which spans approximately 100 megabases. And the same chromosome is also plotted on the x-axis as well, from start to end on the right. And any RNA-DNA interactions identified by a MARGI read pair is added to this contact matrix somewhere corresponding to the origin of the RNA and the target position of the DNA. And if we were to sum up all these read pairs and use this red to white heat map to represent how much RNA-DNA interaction is at each corresponding location of the genome. Here is, and then we derive this contact matrix. And if we were to zoom in to this is about 25 megabases near the end of this chromosome, we see that the RNA transcribed at each row location seem to reach to a neighborhood of the genomic sequence and sometimes can reach several megabase away of its own gene. Coincidentally, if we were to superimpose the known super enhancers in blue stripes, we see that in this case that the RNAs transcribed from super enhancers tend to heavily decorate its large genomic neighborhood. So there is there is a large amount of chromatin-associated RNAs that are transcribed from super enhancers. And through this type of matrix summary, we could start to identify RNA chromatin interactions in cis, where this is mostly from the diagonal, where you see that essentially the target position is not too far away from where the RNA is transcribed from. From this type of analysis, if we were to analyze interchromosomal contact matrix, we will also derive those interchromosomal RNA chromatin interactions or long-range RNA chromatin interactions. So the data analysis pipeline has been packaged into a software tool that doesn't require compilation or tuning, or actually this is utilizing the Docker technology that enables anyone, any computer running on any operation system to directly run this software. So that this will take reference genome, and this will take the IMRG read pairs. Currently, we are also developing a single cell version of this. So there's a cell barcode in there. Take this as input, and eventually it will generate the RNA-DNA interaction pairs and can be plugged into other visualization software. So now, if we were to come back and see what are the chromatin interacting non-coding RNAs, so when analyzing two cell types in this case, the human embryonic kidney and the embryonic stem cells, we see a sizable hundreds of SNO RNA, small nuclear RNA, as well as antisense and pseudogene transcripts are in chromatin-associated RNAs. They seem to decorate the chromatin. And if we ask how far do these genes travel to interact with target genomic sequences, the majority of them, as we were seeing in the diagonal line of the previous contact matrix, the majority of them contact to their genomic neighborhood, forming cis interactions. Still, approximately 20% of the interactions are either long-range interactions or interchromosomal interactions. If we ask, is there any favorable target genomic regions that many chromatin-associated RNA accumulate at? In this case, we plotted all the transcription start sites and their 20,000 bases flanking regions, 10 kb upstream, 10 kb downstream, of every gene in the current genome, human genome assembly. Here we have approximately 34,000 genes, showing as 34,000 rows. And for each gene, for each row, we plotted the transcription start site of the gene at the center, and then 10K upstream and 10K downstream. At the signal intensity here is the cumulative amount of RNA-DNA contacts at that position. So looking across all the transcription start sites of the entire genome, we see that almost always the transcription start site and its immediate genomic neighborhood sequence is where the chromatin-associated RNA is targeting. Whereas if we go further upstream and downstream, there's far less RNA attachment to the upstream and downstream. But most of the observed RNA chromatin interactions seem to accumulate right at the center of the transcription start site. Now we have ordered by the cumulative attachment level at the transcription start site of all the genes. We could ask if this ordering actually order the gene expression level of these genes. For that purpose, for every gene, every row, we plotted its gene expression level by RNA sequencing. So now that the expression level of every gene corresponds to a dot on the same row, the x-axis represents the expression level of that gene. So apparently, we probably wouldn't expect that one indicator would precisely tell me the expression level. Nevertheless, if I were to use a rounding average across genes, we still see a clear positive correlation between how much chromatin-associated RNA is attached to the transcription start site of a gene and on average the expression level of that gene in the same cell type. And if I were to make a quick summary of what we've been spoken, so there are now a few technologies for all versus all approach of mapping all the chromatin-associated RNAs and the respective genomic interaction sequences of each chromatin-associated RNA. And so I described the IMARG technology from my lab that was originally described in this 2017 current biology paper. And in 2019, we improved it into in situ IMARG and described the protocol and data analysis protocol in Nature Protocols. There are now a few other technologies, including grid-seq, char-seq, and most recently radical-seq, which in general uses a very similar idea, but with slight variations in the protocols that attempt to do the same thing. And we put this together in a review article to review these technologies. Coming back to the key questions that we wanted to ask a few years back. First is, what are the chromatin-associated RNAs? Now that we leveraging IMARG, we have seen hundreds of chromatin-associated RNAs, including non-coding link RNAs, small nuclear RNAs, and enhancer-derived eRNAs. And where do these chromatin-associated RNAs bind in the genome? They tend to target transcription start sites of genes. Even if a gene is not activated at this moment in this cell, but there often is already accumulated chromatin-associated RNA on its promoter. And we observed in general a positive correlation of the amount of RNA, chromatin-associated RNA accumulating at the promoter and the transcription level of the target gene of this promoter. So leveraging this information, we wanted to ask if chromatin-associated RNA can contribute to understand a disease generation process. And so we have two questions of strong interest to our lab for several years. And one is why diabetes-induced blood vessel endothelial dysfunctions are irreversible. The second question is how are the fusion RNAs created in cancer? And so I'm going to briefly discuss each case and what we learned there. The first question is regarding the blood vessel. Many of the type 2 diabetic patients suffer from symptoms not directly is per se diabetes, but all types of symptoms arise from different organs. And one of the theme of all those organ problems and failures is their blood vessel is deteriorated. And most apparently is the most inner layer of the blood vessel, which is called the epithelium. This first layer of cells is one cell type. It's a single layer cell type. These are called the endothelial cells, and they form the endothelium. I'm sorry, I said epithelium. It's an endothelium as opposed to epithelium. It's an endothelium formed by a single layer of cells. So these cells are critically important in regulating the perfusion and exchange of nutrients and many molecules inside and outside of the bloodstream. And turn out these endothelial cells turn out to be stressed and become dysfunctioned in diabetic people. So our interest is actually a long-standing question: is how come that so far essentially there's no way to revert, even to intervene the deterioration of the endothelium in diabetic patients. And so in order to investigate this question again, so we collaborated with Dr. Zhen Chen's lab at City of Hope. Here shown Dr. Yijun Luo is from Zhen Chen's lab. And Zhen's lab did two things. One is that they generated a disease model using patient-derived endothelial cells and stress them with high glucose plus inflammatory signal to mimic the environment they would be experiencing in diabetic patients by the diabetic blood. And we assayed these cells in parallel by Hi-C. Hi-C is a technique interrogating DNA-DNA interactions, and by IMRG, interrogating RNA chromatin interactions. And here shows the contact matrix of part of chromosome 2 to part of chromosome 7. We see that there's almost minimal, if any, RNA transcribed from chromosome 2 interacting with any of the chromosome 7 DNA shown in this map in the control normal endothelial cells. As the cells get stressed, after a few days, we see a strong interaction from RNA or a stretch of RNA molecules transcribed from a super enhancer from chromosome 2 to target a genomic region on chromosome 7 near the SERPINE1 gene. So these are the read pairs of the IMRG read pairs, suggesting interchromosome interaction from a link RNA, which actually is embedded in a super enhancer that seem to travel across chromosome to interact with SERPINE1 gene. When we targeted it to degrade this link RNA in the nuclei of endothelial cells, we actually can suppress the expression of the SERPINE1 gene. Turned out SERPINE1 is a key regulator of inflammation that in endothelial cells that is responsible for many of the deterioration and inflammation of the endothelial cells. So at this point, we see a case example where we have an interchromosomal RNA-DNA interaction that underlying at least is part of the seemingly reversible process of endothelial dysfunction. We wish to ask, is this a spatial case or is this a general phenomenon? So for that purpose, we used the same IMRG data to tease out all the other major targets of this link RNA. And we identified, this is on the order of 10 of other targets that were bound in the dysfunctional EC endothelial cells by the link RNA. And we thought we could validate this in the leveraging single cell transcriptome data. So the idea was suggested by a reviewer. The reviewer suggested that if indeed this RNA, this link RNA can activate these target genes from different chromosomes, what you expect to see is that in single cells, you would have more and more single cells in the deteriorating endothelial in the deteriorating blood vessel, and more single cells would co-express this link RNA and its target gene. And it turned out it's indeed the case. So we generated single cell transcriptome from this disease model using comparing both stressed and control endothelial cells. And we also assayed type 2 diabetic people's endothelium and healthy donors' endothelium by single cell transcriptome. So the y-axis represents an odds ratio representing the chance of this gene pair to co-express in the stressed cells or in the type 2 diabetic patients' endothelial cells. So the higher the odds ratio, the more conversion from these two genes being not co-expression to co-expression in the bad condition. And so we see that SERPINE1 happens to be the strongest co-expressing, conforming to the idea that it's under regulation by link 607. A few other genes, every of these genes, let me say, have a positive log odds ratio larger than 1. And in the actual patients, we see a larger variation, but still a lot of the orders were kept. So this suggests, first of all, there is a difference between the actual patient's single cell transcriptome and our disease model single cell transcriptome, but still we see a clear correspondence of these infer interactions. To summarize, we now feel that one of the reasons that contribute to the diabetes-induced endothelial dysfunction that appears to be irreversible is that we didn't know that there are many new RNA chromatin interactions that are formed in the diabetic patient's endothelial cells. And these newly formed RNA chromatin interactions induce expression of a number of inflammatory genes that contribute to many of the desired phenotypes of the endothelium. And degrading these chromatin-associated RNAs inside nuclei actually is a way to intervene with endothelial dysfunction. And so with that, I wish to briefly mention the one other finding that is toward the question of how fusion RNA in cancer are created. So a fusion RNA is a chimeric RNA that came from two premature messenger RNAs. Here shown in this figure, we have premature mRNA transcribed from one gene and another premature RNA transcribed from another gene. But for some reason, there is a fused transcript with part of its exons originated from one gene and part of its exons originated from another gene. It turns out that some of the presence of fusion RNAs or even fusion proteins transcribed from those fusion RNAs are characteristic of subtypes of cancer. For example, this EML4-ALK fusion product actually defines a subtype of non-small cell lung cancer. Not only that, a particular inhibitor developed by Pfizer is approved by FDA around 2011, approved such that such a fusion gene product or fusion RNA product is detected, then this inhibitor can be prescribed. So this is an example where companion diagnostics is approved at the same time with approval of this drug in use in a subtype of cancer. So when we went back to analyze both non-cancer cell line and patient cancer data, we observed from cell line chimeric RNA-seq read pairs linking these two genes, confirming that at least in the cancer cell line, there is formation of this chimeric, this fusion RNA. So in one of our non-small cell lung cancer patients from the tumor biopsy, we observed fusion as well. But from these RNA sequencing read pairs, we can appreciate that although there is fusion RNA between these two genes, both in the cell line and in this particular person, but it is different exons that are fused with the ALK between the cell line and the person. So from normal cells in different cell types using IMRG technology, what we observed is, interestingly, that the RNA molecule of the EML4 gene are attached to the genomic neighborhood of the ALK DNA. So we see, in this case, a coincidence where the fusion RNA's two genes correspond to RNA DNA contacts from one gene to the other. So in our validation experiments based on RNA fish and DNA fish, so we actually validated that at least in this person, there is no underlying fusion gene. So this is a fusion RNA without a corresponding fusion gene. This is a product of trans-splicing. So in this, so our observation is that the gene pair involved in RNA-DNA interaction coincides with the gene pair of a fusion RNA. Our next question is this a special case, or is this a general phenomenon? And to ask whether it's general, Dr. Yan, Zhangming Yan, when he was in my lab, he investigated all the fusion RNAs identified by TCGA from 33 cancer types. I believe the TCGA consortium, when they carried out systematic RNA sequencing from 33 cancer types, they identified a total of approximately 10,000 fusion RNAs. And if we were to plot these RNAs onto the chromosomes with x-axis representing one gene of the fusion, and the y-axis, the x-axis representing the other gene of the fusion. Here is the entire chromosome line versus entire chromosome line. We see that there's more fusion in this region, which spans for several megabases. There are some fusion here as well. If we were to then plot in parallel what is the genome-wide RNA-DNA contact maps in different normal cell types, we actually see a global correspondence between where there's a lot of RNA-DNA interactions and where fusion transcripts in different cancers tend to occur. So with all of this adding together, we proposed a model for fusion RNA biogenesis, and we wanted to call it, we felt it could be called as RNA Poise Model. So the model is the following. So the RNA transcribed from one gene, that's called RNA1, is attached to the genomic sequence of gene 2. And when gene 2 is transcribed, we know that splicing of gene 2 of RNA2 happens most of the time co-transcriptionally. However, at this point, because RNA1 is in its spatial neighborhood, so that it increases the chance of splicing error. And thus, due to splicing error, the two RNA molecules are spliced together to form a fusion RNA. So how can RNA1 in the first place be positioned in the genomic sequence of gene 2? Well, there could be tethering protein, there could be more than one mechanism. One of the possible mechanism is that the 3D genome arrangement already made gene 1's location in the 3D nucleus to be proximal in gene 2 sequence, so that in 3D, these two genes were close together. In this particular case, whenever gene 1 is transcribed into RNA 1, RNA 1 is already in the spatial proximity of gene 2. That case would certainly be the prerequisite of splicing error to happen. That if this is indeed one of the underlying causes, also there are other data showing that when the two pieces of DNA are close in space, there is a greater chances of DNA rearrangement forming fusion gene. And of course, fusion genes could be transcribed into fusion RNA. So this adding together would be a model for us to explain some of the RNA biogenesis pathways to create fusion RNA. So the idea of transsplicing RNA splicing error to create fusion RNA is not new. It has been there for a while. So one of the problems or challenges of this splicing error model is that for a splicing error to happen to splice two RNA together into one, these two RNA molecules has to be close to be spliced together in the first place. So imagine that inside the nucleus, in the space of micron by micron by micron space, the chances for two RNA molecules to meet in that huge space is not great. So that seems to make the splicing error model not easily conceivable if we take the space into consideration. So the RNA Poise model basically says that through RNA chromatin interaction, the two RNAs were close. The soon RNA2 is transcribed. And also that is when most of the splicing events happen anyways. So with that, I have described the work related to RNA chromatin interactions. Are there questions from the audience at this point?
Yes, I have a question. So this is Hyun-min.
Yes, please, Hyun-min.
Yeah. So is there any possibility of having like a ecDNA, like a extra chromosomal DNA, so they have the same, different genes in the same place? They can actually make that happen with RNA interaction or RNA DNA interaction?
That's a terrific question. I would say it's possible. I would say it's possible, and as the previous analysis, we have neglected those small chromosomes. Now that knowing their abundance and importance in cancer, we really are hopeful there are ways for us to find them back.
Thank you.
Hello, Dr. Zhong. Thank you very much for your talk. I have a question regarding this RNA Poise model. So the RNA that is from the RNA 1 that's close to the gene number 2, I mean, we're assuming that that's mRNA, right? It's not a long, like, link RNA or any of that, right? Because I'm asking because—
Hi, John. This model doesn't preclude a link RNA. It can be any RNA molecule.
It can be any. Okay. I was just wondering, and then do these genes that present this situation, these cells that present this situation tend to have mutations in the spliceosome-related genes, or why wouldn't we see this in the regular?
So there were previous studies that suggest that errors in RNA splicing doesn't require spliceosome mutation. It is just an error. So when splicing happens for a lot of times, in a small chance, splicing error will happen. However, if cells are stressed, the frequency of splicing error increases. So that mutation in the spliceosome genes is not so far, most data point it's not a prerequisite. And so for splicing error to happen.
Thank you.
So thank both for the questions. So I have a question for the audience as well. So what are the photographed materials here in this picture?
Yeah, my favorite is socks.
Yes, socks.
So microscopically zooming in, that appears to echo both Rong's and Sue's identification. So with the last few minutes, I wish to switch gear to discuss RNA on cell surface. Is that a type of cell surface molecule? So what I hope to discuss is a way of naturally happening display of the nuclear encoded RNA from the nuclear genome, transcribed from the nuclear genome, that is presented and stably associated on cell surface and its impact on cell-cell interactions. So the first question is, does such cell surface RNA exist? First of all, cell surface is apparently critical for cell environment and cell-cell interactions. So the RNA transcribed from the nuclear genome is generally not expected to appear on the surface of eukaryotic cells, at least the cells with intact cell membranes. And so here I wish to define what are precisely the RNA species I wish to discuss. So we define this maxRNA, short for membrane-associated extracellular RNA, as nuclear genome transcribed RNA that are stably associated with the surface of the intact cells and exposed to the extracellular space. So our goal here is to test for the presence of maxRNA in mouse and human cells, and to test if any maxRNA can influence cellular functions. We were not the first group who suspected the plausibility of maxRNA's existence. It turned out that in the 1960s and the 1970s, a pioneer cancer biologist, his name is Leonard Weiss, had a similar suspicion, and he actually carried out some experiments to test it. So in the 60s, it was already known that cells undergoing metaplastic transformation, that is like a transdifferentiation or cancer generation process, those cells, their cell surface tend to be even more heavily negatively charged. Like cancer cells tend to be even more negatively charged to their plastic precursor. So in general, this negative charge were attributed to more glycans due to glycolysis. However, Leonard Weiss felt that, well, RNA are also negatively charged. He asked whether, in addition to the sugar groups, whether RNA may also be present in cancer cells' periphery. So at that time, what Leonard did was that he used radio labels to label intracellular RNA that at least he thought was intracellular RNA. And he actually was able to identify on the cell periphery those radio labels. In addition, he found those peripheral labels were sensitive to RNA's digestion. So therefore, in Leonard Weiss's paper in 1966, he wrote, these results are interpreted to indicate that RNA is a structural component of the peripheries of these cells. So around his time, his papers were seen and cited by other scientists. For example, Sarah Zalek studied another metaplastic transformation, a transdifferentiation process, where Sarah actually did similar experiments and reached similar received similar data. Although at that time, Sarah perhaps was not completely ready to take Dr. Weiss's conclusion, and Sarah wrote, "The nature of this material is currently under investigation," while citing Weiss's paper. So these pioneer studies were not followed up for almost half of a century. So our goal as an approach as follows. First, we wanted to start from a lymphoma cell line to test if Weiss's reported material are indeed RNA. And second, we wanted to test in primary human cells if there is any cell type specificity and what cells present max RNA. And thirdly, we wish to identify what specific max RNAs are on primary human cells. And finally, we wish to test if a specific max RNA mediate cell-cell interactions. For the first question, how do we test if this potentially peripheral structural material of lymphoma cells are indeed RNA? So we leveraged nanotechnology called membrane-coated nanoparticles, MNCP, developed by my colleague Liangfang Zhang's lab here at UCSD. What this MCNP does is that Liangfang's lab tease apart cell membrane and wrap it tightly around a nanoparticle core, and thus forming a designed core that is tightly wrapped by cell membrane. There are two unique features of this technology make it quite useful to us. One is that the inside-outside orientation of the membrane is kept, so that on the nanoparticle core, the inside is still facing inside of the membrane, the outside of the membrane is facing outside. Second is that any intracellular material is rigorously removed during this process of assembling such nanoparticles. So leveraging this technology, we developed a technique which we call a surface-seq, essentially that first construct MCNPs from lymphoma cells, just using the lymphoma cells membrane, and then purify and sequence any RNA molecule facing the outside of such MCNPs. So we developed two technical variations of this sequencing technique, and I'm not going to go into details. So applying these two variations of surface-seq to lymphoma cells with different repeats, we've identified 92 non-coding RNAs that consistently reappeared in every of our biological repeat. So MALAT1 gene and MALAT1 RNA is one of the 82 candidate surface RNAs. If we were to plot the 5 biological replicate from 2 technical variations of these surface RNAs reads onto the MALAT1 gene, we see that not everywhere of the MALAT1 RNA seems to be detectable on the cell surface, whereas this part of the subfragment of the RNA seems to be always presented in every biological sample of ours. So we wanted to use RNA fish to test if indeed this part, this fragment, RNA fragment, is presented on cell surface. So our single molecule RNA fish seem to actually detect some of the signals on the cell periphery, and in our single base mutant control probes doesn't show any signal. So this also we can use some live cell imaging techniques to test these cells are indeed live cells. And so in summary, what we find is that RNA is indeed present on leukemia cell surface. And because this is RNA sequencing, we feel that the evidence for this material RNA are much stronger now. Our next questions are, is RNA present on normal primary cells from humans? And do all cell types present surface RNA? So in order to test whether RNAs are present on normal primary cells, we needed a way to crudely label the cells that may present surface RNA. And we developed a technique which we call in situ surface fish. What we do is that we generated a random nucleotide library, and we incubate this random library of fluorophore labeled probes with primary human cells. And we watch the cells and we image them. And then we use these probes to pull out those cells for further analysis. If after wash, if we were to image these fluorophores with, and also image with antibodies against different subtypes of PBMCs, we see co-localization or co-appearance of CD14 surface protein and putative max RNAs on the same cells. So if we were to quantify this, in order to quantify this, we split the PBMCs into two pools of single cells. One pool are what we call IS-fish plus. Those are the ones that are pulled down by FACS separated with positive fluorophore signals. And the rest are the negative, which presumably doesn't present any putative max RNA signal. And then we subjected these two pools of cells to single cell sequencing. And a t-SNE plot seemed to separate these two pools of cells, which is desirable to us. Then if we were to plot the expression levels of marker genes of different PBMC subtypes, here we are plotting in this figure showing two marker genes of monocyte. We see that these marker genes seem to concentrate to the single cells of the IS-fish plus population, which coincides with our microscopy observation, that is, the monocyte seems to be a major surface RNA presenting cell type in PBMC. To summarize, for the time being, cell surface RNA is present on normal primary cells. Not all cell types present cell surface RNA, at least to our detection limit. And the cell surface RNA is present on CD14 plus monocytes. Our next questions are, what genes give rise to the primary cell surface RNA? And does any cell surface RNA modulate the functions of their primary cells? And to identify what genes give rise to surface RNA, we combined IS-fish to sequencing. We essentially purify those RNA by the antisense probes library, and then sequence whatever co-purified RNA. And in order to minimize background noise, we actually generated 3 technical variations, each adding some additional step to hopefully narrow down the RNAs that are indeed either monocytes or on cell membrane as additional filtering steps. And in the end, we derived a few hundreds putative candidate max RNAs. And we tested around 11 of them. Most of our tested candidates are in the intersection of the 3 technical variations. So how do we test whether a putative cell surface RNA may have any impact to the cell's function? First of all, among all the possible cell functions, we feel that cell-cell interaction is where we are going to test. So one thing that we know about monocyte is that one of the critical feature of monocyte is that it would adhere to the endothelium. We talked about endothelial cells of the blood vessel. So the monocyte would adhere to endothelial cells, and the adherence is a prerequisite step for the monocyte to migrate through the endothelium and transform into macrophages. So this attachment is actually a quite well worked out quantifiable assay between monocyte and endothelial cells. It's being used actually widely as a way to interrogate cell-cell interactions. So monocyte endothelial attachment assays is standard in research of endothelial cell and monocyte functions. We wanted to leverage this and leverage the quantifiability of the attachment levels. So what we do is that we incubate the monocyte with antisense probes of every candidate max RNA, every candidate cell surface RNA. So we designed antisense probes against every of the 11 putative surface RNA, and we incubate such antisense probes with monocyte for a very short amount of time, for only 1 hour, and then we carry out we culture monocyte EC and carry out the standard monocyte EC attachment quantification. And on the y-axis are the quantified monocyte EC attachment levels. As you see, some of the controls, as expected, control probes do not change the attachment levels. Several of our candidate max RNA may not have a significant change, but at least 2 max RNAs, when we interfere with the extracellular in the extracellular space, they seem to strongly suppress monocyte EC attachment levels. So at this point, we feel that there's data suggesting some surface RNA can modulate monocyte EC attachment level. Our next question is, does this modulation require the entire RNA sequence or not, or only a piece or fragment of the RNA? So if I were to reinvestigate this MNDC3B gene, so here is this gene on the genome. Here are the exons and introns. The red tracks shown here are our IS-FISH sequencing data, and you see reads are aligned to different parts of the gene. There are a lot of reads aligned to the 3' UTR of this gene. And the blue tracks are our different controls. As you can imagine, there's only a tiny amount of cell surface RNA, even if there is any, so that it's important to contrast to various types of controls. In every our technical variation, we have at least three control replicates. So we then designed individual antisense probes targeting different parts of this gene. And we had 5 probes targeting different parts of the 3' UTR of the gene, because there simply is more surface sequencing reads at the 3' UTR. And then we quantified the monocyte EC attachment level from extracellular incubation for 1 hour of every antisense probe. So what we see is that the control didn't change. The oligos targeting some of the exons don't seem to manifest anything. Targeting the 5' end of the 3' UTR didn't seem to work. But the more toward the 3' end, then the larger the impact is to change the monocyte's ability to attach to EC. At this point, we feel that not all parts of at least the mature transcript of the max RNase gene are equally important for their cell surface functions. So if I were to summarize, so this technique, surface-seq, can identify the genes of primary cells max RNA. And what we have discussed so far, we feel that we have tested Leonid Weiss's suspicion. Lymphomas cells do present max RNA on their surface, and it's verifiable by both sequencing and RNA-FISH. And in primary cell population, monocyte is a major type of max RNA presenting cell type in human PBMCs. And blocking specific monocyte max RNA can reduce monocytes' ability to interact with endothelial cells, and blocking extracellular blocking of just a fragment, a particular fragment of particular putative surface RNA can reduce the monocytes' ability already. So with that, I wish to mention the people behind the scenes. The Norman Huang, Dr. Huang, he is the one, the main person who generated the data I described about max RNA with the help from Xiaochen Fan, Cassia, Zaleta Rivera, Chui Nguyen, and Zhang Mingyan. So Zhang Mingyan, I mentioned him several times. So he is the one behind analysis of fusion RNA, and also behind the analysis of the surface RNA data. Dr. Liangfang Zhang's lab provided the critical nanoparticle technology for us to derive surface-seq technique, and Dr. Zhen Chen's lab provided endothelial cell expertise. With that, I wanted to open for any questions.
Yeah, thank you so much, Sen. This is super interesting biology, and actually many pieces of super interesting biology there. So Ahmed somehow has internet problem. He's not able to join again. So now if anyone has question, you can either unmute yourself or you can use the chat box to put your question there.
Hi, thanks for the talk. Super interesting. I have a couple of questions. Two of them are sort of linked to each other. I was wondering if you could talk about your thoughts on what's the mechanism of RNA getting onto the cell surface. Like, is it from inside the same cell? Is it somehow picked up from the environment? And then, like, potentially a related question, what makes you think, like, why monocytes?
Yes, excellent question. So first, how is RNA get onto cell surface? So if I were to forget about the human genome transcribed RNA, for viral RNA, it's known to actually can get attached to cell surface. In fact, some of the toll-like receptors are meant to target viral RNA, and as a way of actually by stabilizing and interacting with viral RNA, so the viral RNA are attached to the surface of some cells. So here the interest is to discuss how about human genome produced RNA rather than alien invasions. Our initial inspiration came from bacteria. In bacteria, for how bacteria use a relatively small genome and allow for their transmembrane protein to localize to the cell membrane. One popular way is that after translation, the bacteria messenger RNA is not dissociated with the translated peptide of a transmembrane protein. Instead, the still connected messenger RNA actually contains localization signal for other protein to pick up and translocate the RNA peptide chimera to bacteria membrane. And then, of course, the protein peptide is needed to penetrate and stabilize the transmembrane. So in bacteria, RNA is a localization signal to get across the membrane, to get to and across the membrane. And a few years back in eukaryote, there was one example of a messenger RNA not dissociated and used as localization signal to penetrate, at least to bring to and let the peptide to penetrate ER. Although there's not yet a worked out example for penetrating the of the human cell plasma membrane, but a similar mechanism is described in one gene and one RNA actually is the localization signal to bring the protein across ER membrane. So that makes us to feel that perhaps it's just that we haven't found it. Perhaps this nice way of localizing transmembrane protein, perhaps human genome still uses it. Coinciding with this idea, although we haven't worked out the mechanistic steps, but indeed, if we analyze what are the gene transcripts in our PBMC's cell surface RNA, it's heavily associated with transmembrane proteins messenger RNA. So that coincides with our initial suspicion that perhaps the same mechanism is still used in humans. It's just that we need to work out all these actual steps of a specific example. The other possibility that I would not rule out is just like any of our endocrine or paracrine signaling, right? So for other ligands that are stabilized on cell surface from endocrine or paracrine signaling, we know that the ligand can be produced by another cell. So that means that we probably, at least conceptually, it's possible that RNA transcribed from a different cell potentially can be exposed and somehow, just like a ligand, get stabilized onto another cell's surface. This process may or may not require the original cell producing the original, let's call it ligand RNA, to be dying a lot or not. I can imagine a healthy cell somehow deposit extracellular RNA as well as a dying cell. With respect to a dying cell, there is a process called phagocytosis, mediated by macrophage, monocyte, and T cell. These cells tease apart a target cell and take some of the cellular content of the cell under attack and present it on the host cell surface. So this attack, take, and display process is, I believe it's called phagocytosis. So that's a process that's worked out for intracellular proteins for most of the time. We imagine that it's possible that a similar process is not only for display of the cell being attacked, dying cells intracellular protein. Perhaps it's the target cell's intracellular RNA or fragments of RNA could be displayed in a similar manner. This goes back to your question about why monocytes. While we are pursuing the possibility, at least the current data is more like the monocyte displays on its own. But monocyte also is a cell type participating in phagocytosis. So there is a chance that through the similar phagocytosis process, it displays genes transcribed from other cells. So those are our suspicions.
I think that's actually the question I want to ask as well. And thanks, it's kind of answer majority of my question as well. But I think on top of that, I'm thinking, so if you can do something like a RNA pull down, RNA protein complex pull down, something like a chip sequence for RNA, you know what? So how the RNA is anchored on the cell surface? You know what is the sort of the partnering protein or other entity there, and kind of better understand the mechanism. That's what I'm thinking. So the second is when you were talking, I also suspect that I think that's some kind of phagocytic activity that might be associated with this. And have you looked at kind of more mature kind of phagocytic cells like a macrophage versus the monocyte? If you differentiate the monocytes into macrophage, do you see this kind of max RNA presentation on the surface significant upregulated?
This is a wonderful idea. So we haven't really we really haven't matured those, but in line with your idea, we did a simpler experiment. We use LPS to stimulate stimulate PBMC. Yeah, so we see that the surface, I call it surface RNA. Now, it should be cell-peripheral RNA by our enrichment method. A special class that seem to be LPS activation inducible from PBMC. So that is the preliminary data we have towards that line. But I agree with you that if we can mature monocytes, that perhaps it would be a cleaner experiment.
Yeah, but I think a lot more things no one knows. So it's a completely new field you opened up, honestly. So this is super cool. So I have another question back to your earlier kind of histone RNA interaction. So any way you can kind of verify at a functional level, for example, like a shRNA or siRNA to kind of silence that particular piece of the sequence, kind of interact with the histone.
So yes, so to the histone, the RNA histone. So the RNA chromatin interaction. So we indeed, I didn't talk about that experiment. It's an experiment done by Zhen Chen's lab. So she designed LNA to specifically degrade inside the nucleus this link 607 transcript, and she actually, her data showed that when this specifically nuclear degradation of this link RNA actually suppress the otherwise stress-induced expression of the SRP1 inflammatory gene, but cytoplasmic degradation does not have a similar suppression. So one of the challenges that we faced in trying to use interfering or lockhead nucleic acids to degrade the putative regulatory RNA is there's always a fear that we have interfered with the translation of that RNA. So that RNA, if its own has a translation product, then if we knock it down, then what are we testing? Are we testing this RNA's own regulatory function, or are we testing the protein product of this RNA? So that's why we actually chose this not yet well annotated link RNA that happens to be transcribed from an enhancer. So that was in order to minimize the chance that we interfere something else. So similarly to the surface RNA interrogation, right? So if we were to do any genomic perturbation or intracellular inhibition of this RNA, so there's always a fear that whether the intracellular protein get perturbed in that way. So that so far we are choosing extracellular perturbation.
Yeah, I think for the interest of time, there's one more question in the chat box. Maybe I think from Constantine. If you can unmute and ask, yeah, please go ahead. Otherwise, I can read for you.
No, all good. Yeah, so I think just one, going back to the extracellular membrane localized RNA. So if the hypothesis is that mRNA can potentially serve as a localization signal, would it be a testable hypothesis that like fibroblasts pumping out a bunch of ECM proteins would also be expected to be enriched for max RNA as well?
Yes, I feel that's an excellent hypothesis, and I like fibroblast because it's like so for some reason also for we also tested different cell types for just like a crude assay of whether they may present surface RNA. It looks to us that more terminally differentiated cells are less likely to have detectable. So this is in addition to, yes, they have fibroblasts pump out things that, but also fibroblast is perhaps not a terminally, so that there's just more. We tested more than 10 cell types, and I don't recall fibroblasts in per se, but in general, they not terminally differentiated cells appear to more likely to have.
Yeah. No, thank you.
Yeah, thank you very much, Shen. It's a fantastic talk. Basically, 3 fantastic talks combined in one. So I think also this is a kind of perfect talk to conclude our series 4. We're going to have Series 5, but Ahmed is working on a schedule. So we already have people from, I think at 10X reach out to me. They are interested in speaking again, and NanoString as well. And I think Jim Fan from Johns Hopkins. We have 4 or 5 already lined up, but if anyone interested in speaking, please don't hesitate to email me or Ahmed. So thank you very much, and hope you guys have a wonderful weekend.
Thank you.
Thank you, Ron. Thank everyone.